Hey everybody, Dr. Duncan here with a video lecture for BI 225 students on cladogenesis and phylogeny. A common misunderstanding is that species descended linearly from their ancestors, but this ignores the fact that there are lots of other related species to take into consideration. Just like in a family tree, there is branching, and to look only at the direct line would mean that you are ignoring important relatedness. Consider this diagram illustrating the evolution of horses. There's some fascinating patterns here. Earlier horses were smaller, had many toes, and based on their dentition, lived in a forest environment. As the environment got drier and grasslands expanded, horses evolved to be larger, they ate more grasses, and they had fewer and fewer toes, which facilitated faster running in open habitats. Also notice that most of the branches on this family tree stop before reaching the modern era at the top of the page. Those are lineages that went extinct. Obviously, there's a lot more to know about horse evolution than a simple tracing of its direct ancestors. Understanding the evolutionary relatedness of organisms is central to much of evolutionary biology. There are several closely related subdisciplines or practices that you will hear about. These include systematics, which is the study of diversity of life and evolutionary relationships; taxonomy, which is the process of identifying, naming, and classifying species; and phylogeny, which is determining the evolutionary relationships of species. It's this last category we're going to be focusing on during today's video lecture. In particular, we're going to spend a lot of time looking at phylogenetic trees, which are hypothetical relationships of evolutionary pathways. It's important to practice being precise and accurate with your description of how species evolved in relationship to one another. Let's take a look at this human evolutionary tree on the left. Like the horses, we see that it is similarly complex. There's many extinct lineages. But it is not correct to say that we evolved from Neanderthals, as you can see in the design of the phylogenetic tree on the left. That's a commonly misunderstood concept. Or it's similarly incorrect to say that we evolved from apes or monkeys, as you can see on the right. We share common ancestors with other hominins, other earlier humans, and we share common ancestors with apes and monkeys. That is the correct way to talk about our shared common ancestry. This is one of many diagrams to represent the tree of life. It beautifully shows the scale of Earth's biodiversity, the relatedness among major taxonomic groups, and where we humans fit into all this. This particular tree of life is not intended to be used as a scholarly reference tool or as a complete picture for Earth's evolutionary history. For example, while 99% of species that have ever existed on Earth are now extinct, only a very few extinct main branches are shown. Instead, this diagram illustrates a great lesson of evolution, and that is that we are related not only to every other living thing, but also to everything that has ever lived. Another flaw in this and similar diagrams is that the spacing of the branching pattern suggests a regular splitting of lineages through time. In the PowerPoint that is associated with this video, zoom in on the illustration and you can see what I mean by the regular spacing events, which of course are not realistic. This is a much better tree of life, though it doesn't really look like a tree anymore. This one shows extinctions and relatedness much better. But it still isn't complete. There is simply no way to map 50 million species in full detail and full accuracy with simple visual models such as this. Where are humans, by the way? We're that inconspicuous branch at the lower right-hand corner with other eukaryotes, indicated by the red circle. Here's a third and our final example of the tree of life. This is a much more fun one. This is a screenshot from a website called onezoom.org. This is one of several uses of computer technology to illustrate the tree of life in ways that are better than a two-dimensional representation. Pause this video and take a few minutes to explore onezoom.org. The image is linked to the website so that you can visit using the PowerPoint file posted in Moodle. Just navigate to this slide and click on the image. So why should anyone care about phylogenies? Well, firstly, there's something to be said about how understanding evolutionary history can enhance one's appreciation for the wonder and beauty of the human story and the story of life on Earth. But more practically, since many of you are headed into healthcare, let me paraphrase what Dr. Neil Shubin said. He said that if you want to understand why the human body is the way it is, then you need to peer into our evolutionary past. I encourage you to watch him talk about his research on the Colbert Report. The link is on this slide, which you can find in the PowerPoint file posted on Moodle. And if you ever have time, his book, The Inner Fish, is a great read. It'll be a page turner for anybody with biological background. Okay, it's time for us to learn how to read phylogenetic trees. Whenever you see a tree, the first thing to look for is the direction of time. In the examples that'll be on this slide, time goes from the bottom towards the top of the diagram. Sometimes you will see trees displayed such that time goes from the left to the right or the right to the left, and sometimes it'll go from the top to the bottom. A second thing to notice is the branching patterns on the trees. Each split represents a speciation event, where an ancestral lineage diverged into two or more new species. At this point, you should be thinking about the allopatric and sympatric speciation events. Third, note that living, surviving species are represented at the tips of the tree. In these examples, the species are represented by numbers at the top or letters in the bottom tree. Now, once you understand all this, you can use these trees to study the hypothesized relatedness between species in a particular phylogenetic tree. In the bottom one, you can see that species B and C share a common ancestor with species A. The last point in time when all three species shared a common ancestor is at the branch point labeled with the orange box and text. Similarly, the last point in time when species B and C shared a common ancestor is at the point labeled with the blue box and text. Finally, check out the green box on the branch leading up to species C. That part of the branch indicates the final stages of the lineage that becomes that species. Any of the positions along that last branch towards the modern species C is a unique ancestor shared only with the modern form of species C. There's a couple more things to learn about how to read phylogenetic trees. The vast majority of species that have ever lived are no longer alive today. Such species are said to be extinct. These are represented in phylogenies by branches that do not reach the end of the timeline. Three such lineages are in this diagram. Species that are alive today are known as extant species. Extinct versus extant, the words are similar, so find a way to not get them confused. Another important concept is the clade. Species that share a common ancestor are known as a clade. In this diagram, we could correctly call the beetles a clade. These are the species in the blue circle. Clades can be nested. Notice that the beetles are one of several clades comprising the insect clade, which is circled in orange. And the insects are in a clade nested within the arthropod clade, which is not fully displayed here. Evolutionary biologists spend a lot of time creating phylogenetic trees by measuring and comparing traits of living and extinct species. As you will learn and practice in the semitext exercise Flowers and Trees, traits or character states are mapped onto the tree at approximate times that they arose. They can occur just before branching points or along the straightaways. Traits are said to be ancestral if they are possessed by ancestral individuals in the tree. Traits are said to be derived when they are possessed only by descendant species. Ancestral traits are also known as synapomorphies, while derived traits are known also as apomorphies. It may help to remember that the Latin prefix syn or syn means together, as implied by words such as synchrony or synthetic. The Latin prefix apo is less common but means away or apart. In the example on this slide, petal tips is a synapomorphy for peak 1, 4, and 3. Spur bottoms and petals are apomorphies because they arose later in evolution. They are more recently derived. Anthers are an ancestral synapomorphy shared by all four species. This is an example from Simutext, so if you haven't done that exercise yet, this will make sense to you after you've done that exercise. In a previous class, you learned about how a species can split into two species by way of allopatric or sympatric speciation. The creation of new species by way of splitting is known as cladogenesis. However, not all new species arise through cladogenesis. Lineages continue to evolve regardless of whether or not it ever splits. Sometimes we see in the fossil record where a lineage doesn't split for a long period of time, but the species does evolve as it continually adapts to its environment. This process can lead to enough evolutionary change that biologists designate as a new species the more recent forms of the lineage. That process of new species formation without splitting is known as anagenesis. A comparison of cladogenesis and anagenesis is depicted in this diagram. Usually anagenesis is not drawn on phylogenetic trees, but there are exceptions. Okay, that covers the basics of how to read phylogenetic trees. Let's quiz your comprehension. Pause the video and evaluate the veracity of the above statement. I'll tell you the answer next. Okay, this statement is false. Notice that marsupials share a common ancestor with placentals more recently than they do with the monotremes. That means that marsupials are more related to placentals than to monotremes. Did you get confused because all three groups are equidistant at the tip of the tree? Don't feel bad, that's a common mistake by newbies. The way to determine degree of relatedness is by looking at the branching pattern, not by the order of species listed at the tip of the tree, nor by the distances between the tips of the branches. Always look at the branching pattern to determine relatedness. Okay, let's quiz you a bit more. This is a phylogenetic tree of 12 species of fruit flies. Examine the branching pattern and the timeline at the bottom of the tree and answer the question. Your choices are at the right. Pause the video now and figure out your answer. Okay, the correct answer here is C, which is 25 million years ago. Okay, here's another question. Pause the video again and derive your answer from the choices at the right. Okay, the correct choice here is A, Simulans and Erecta. These two are the most closely related because they have the most recent common ancestor. Did you get confused because of how the names are at the end of the timeline? Remember, relatedness is measured by when species shared a common ancestor. Simulans and Erecta diverged from their most recent common ancestor at about 10 million years ago, much later than the other two pairs. Okay, here's a quick review of some of the things we've learned so far. Multiple branchings from a common trunk to show relatedness among species is the way that phylogenetic trees work. Organisms are related to one another through descent. These are hypothetical relationships that are depicted in phylogenetic trees. Closely related species are assumed to share more characteristics because of common descent. Shared derived traits are known as synapomorphies, while a derived trait that's not shared is known as an apomorphy. A quick word about cladograms versus phylogenetic trees. Sometimes evolutionary biologists create cladograms, and sometimes they construct phylogenetic trees, also known as phylograms. The image at the right shows the differences between the two. What they have in common is that they show hypothesized relationships among organisms based on similarities and differences. Cladograms show relatedness between objects, any objects. For example, you could look at the relatedness between different types of cars. based on how they're built. Cladograms are built using the presence or absence of shared traits. The branching pattern in cladograms does not necessarily represent the total degree of change as species are evolving. They might just show one trait, for example. Nor do cladograms necessarily represent how much evolutionary time has elapsed. Phylogenetic trees, on the other hand, always show the amount of evolutionary change relative to the amount of time that has gone by. Unfortunately, both terms are used interchangeably by biologists, and that makes it very confusing. You have to look at the information presented to know whether it is a strict cladogram or a phylogenetic tree. So, how are phylogenetic trees built? First, let's review what is a clade. Recall that a clade is a group of all organisms that share a common ancestor. As illustrated here, a clade can be as small as a single species, or it can include many species. Now let me also introduce to you a new term, monophyletic. A monophyletic group is a group of organisms descended from a common ancestor or ancestral group. For our purposes, the term clade is used the same as we use the term monophyletic group. One of the strategies for building phylogenetic trees is to look for homologous structures. These are organs or body parts that appear in different species and are similar in structure and location, but they don't necessarily share the same purpose. A classic example is with the mammalian forelimb. In the illustration, we see how through evolutionary time, the radius, the bone in orange, has been tweaked by natural selection to have very different functions. Early biologists who figured this out had noticed structural similarities in the forelimb bones of mammals. With this, in comparisons of other organ systems, they were able to deduce a hypothesized evolutionary relationship between these and other mammals. The hypothesis is that the same bones were present in the forelimb of a common ancestor to all these species, and that through natural selection the forelimb bones were re-engineered for many different purposes. Here at BSc, we teach an entire course about this known as comparative vertebrate anatomy. Grouping organisms into clades based on shared or absent traits can sometimes get tricky. For example, this phylogenetic tree shows what we know about tetrapod evolution. At first, everything might look fine. Here are the mammals, here are the bony fishes, and here are the amphibians. Each is a tidy clade. But check out the so-called reptiles. Any grouping of what we traditionally call reptiles should also include the dinosaurs. However, if we include dinosaurs, we leave out the birds. Clearly, based on these evolutionary relationships, the reptile clade should include birds. But as we all quote-unquote know, birds are birds and reptiles are reptiles, right? This illustrates a problem that appears sometimes when biologists try to construct phylogenies. In this case, terms derived long ago and that are in widespread usage, such as bird and reptile, do not reflect the true evolutionary relationships. The group of organisms we call reptiles are an example of what's known as a paraphyletic group. This is a group of organisms that is an incomplete evolutionary clade. Because birds are more related to dinosaurs than any other group, yet dinosaurs are commonly referred to as reptiles and birds are not considered reptiles, this is a paraphyletic group. The opposite problem is that of a polyphyletic group, seen in the lower left. This is a grouping that includes all of one evolutionary clade and part of another. It might help to recall that the prefix poly means many, and a polyphyletic group has components of more than one clade. Okay, let's check in to see if you are understanding these concepts. Pause this video to answer the question. The correct answer is B. Because a polyphyletic group has members from more than one clade, this group will have the least amount of shared characteristics. Here's another challenge. Pause the video again and answer this question. The correct answer here is C, polyphyletic. Both bats and birds have wings, but they are not in the same clade. And here's one more challenge. Pause the video to see if you can answer this question correctly. Here the correct answer is B, paraphyletic. Dinosaurs are all in the same clade, but their evolutionary clade includes birds. Because the clade is incomplete, dinosaurs, as a concept, are a paraphyletic group. Okay, let's get back to understanding how phylogenetic trees are built. Trees are often built by examining synapomorphies. Remember that term? Synapomorphies are traits shared by two or more organisms. Note the chart below, which lists the 5 species depicted in the cladogram above, and whether they have each of 6 traits. Go ahead and pause the video to look over the chart and the cladogram. Now, compare the rabbit and the lizard to all others. In this case, they have 4 synapomorphies, which is more than any other possible pairing. Thus, they get grouped as a clade because it is highly likely they share a common ancestor more recently than any other pairing of species. The one trait they do not share is mammary glands, and this is an apomorphy specific to the rabbit. The cladogram is constructed using the process of grouping species based on the number of synapomorphies. Another strategy of constructing phylogenetic trees is to use the principle of parsimony. This is making decisions about evolutionary relatedness based on prioritizing the most simplest of explanations, and that would be the explanation that involves the fewest number of evolutionary steps. Check out this simple phylogenetic tree for 5 species of vertebrate. Notice the 5 traits used to make this tree and how they are mapped onto the tree. If you count them, you see that only 5 evolutionary steps are needed to explain this hypothesized configuration of relatedness. Now, check out the hypothesized tree on the right. Same species, but this tree shows the hypothesis that the sea bass and the lamprey are a clade. For this to have occurred in evolution, the jaw would have had to have evolved twice. Once after the sea bass's ancestor split off from the lamprey, and once after the ancestor to the antelope, eagle, and alligator split off from the ancestor to the lamprey and the sea bass. This scenario at the right involves 6 evolutionary steps. Because the scenario at the left is more parsimonious, it is the favored hypothesis for the relationship between these taxa. Does this mean that that evolution always follows the most parsimonious path? No. Sometimes similar traits really do evolve more than once. For example, consider the patterns of mimicry we see among some groups of animals. Evolutionary biologists work through these challenging histories by mapping many traits. The more traits that are mapped, the more likely the resulting phylogenetic tree will be the correct hypothesis. Another clue that evolutionary biologists can use to examine relatedness and develop phylogenetic trees is the presence of vestigial structures or traits. These are traits that are still present in some form, but that have lost most of their ancestral function. For example, snakes still have remnants of a pelvic girdle, and boas, a type of snake, have vestigial claws of a hind limb, as seen in the photograph. In case you were wondering, there are several features in humans that are thought to be vestigial structures. These include the coccyx, the appendix, wisdom teeth, a remnant of a third eyelid, and ear muscles for orienting our hearing. One thing that can throw off investigators are analogous structures, also known as homoplaces. These are traits that are similar but evolved independently in evolution. Usually the trait evolved because organisms were adapting to similar environmental conditions. For example, sharks have pectoral fins, penguins have flippers derived from wings, and dolphins have a flipper evolved from a foreleg. Each of these structures helps with swimming and is similar to the others. But they evolved at different points in evolutionary history. Now, no biologist today would come to the conclusion that penguins are more related to dolphins or sharks than they are to other birds. But there are other groups of organisms for which it can be very difficult to determine the evolutionary relationships. And homoplaces can really throw off investigators from figuring out the right relationships. Homoplaces are the result of what's known as convergent evolution, where distantly related species evolved traits that are very similar to one another. Usually, it is because these species live in very different environments and have similar selective pressures on them. A vivid example of this is the contrast between marsupial mammals, which are primarily found in Australia, and the placental mammals, which are those found throughout the rest of the world. Both groups have evolved some remarkably similar traits, even though they haven't shared a common ancestor for 160 million years, which is, even by evolutionary standards, a long time. Remember that phylogenetic trees are hypotheses that can be tested. For example, based on fossil evidence, it has been hypothesized that cetaceans, the toothed whales and the baleen whales, they evolved from a terrestrial vertebrate, something akin to the modern cow or hippo. Whales have a vestigial pelvis, which is one of several lines of evidence supporting this hypothesis. The cladogram at the bottom right of this slide shows that if this hypothesis is true, the early ancestors to modern whales should have an ankle structure known as a double pulley. We can't find that structure in modern whales because it was either never there or because whales lost this structure over time. Recent fossil discoveries have confirmed the prediction that ancestral whales had a double pulley ankle. Paleontologists found ancient whale fossils with hind legs and pelvises. These whales had the same kind of double pulley ankle bone that modern antelope, camels, cows, and hippos have. This was yet more and very strong evidence that whales have a terrestrial ancestor. One final point I wish to make is that this video lecture is just a sampling of the many techniques that scientists use to develop hypotheses about the relatedness of species. Many of the most influential techniques today involve directly comparing the genetics of species to measure their relatedness to each other. Okay, that's it for this video lecture. If anything here was unclear, cross-reference it with your readings or other videos and contact your instructor.